we can see about the structure of nucleus in this topic. The nucleus was first discovered by Robert Brown in the year 1831. Later, the material which is present inside the nucleus was given the name chromatin by Fleming. He gave the name chromatin because that material was able to stain well by any basic dyes. Okay, when we add such a stain or any dyes, it will take up the stain effectively and it can be seen as colored material. That's why it was named as chromatin by Fleming. Okay. Now when we see the interphase nucleus, what is that interphase stage? It is the phase which we can say it as non-dividing phase of cell. What is non-dividing phase? When a cell is not dividing, it will be in a phase called the interphase. Okay. So we can say it as the non-dividing phase of the cell. So in that interphase nucleus, if you see, we can see three main parts like one is the highly extended and elaborate nucleoprotein fiber called the chromatin. Okay, so here you can see the fibers. Can you see it? A network of fibers are there. Highly extended, very long and elaborated fibers are there and it is called chromatin. Then the matrix which is present here, matrix means the fluid which is present inside is called as the nucleoplasm, and one or more stained bodies will be there and it is called the nucleoli. Okay, so these are the three main parts of the nucleus. So what are they? One is the extended and elaborate nucleoprotein fiber called the chromatin. Another is the nuclear matrix called the nucleoplasm, and next is the spherical bodies, one or more spherical bodies called nucleoli. So these are present inside the interphase nucleus. Okay, nucleoli is a plural, nucleolus is a singular. Okay, now when we see this nucleus under the electron microscope, we can see that this nucleus is surrounded by a nuclear envelope, and this envelope consists of two membranes which are arranged parallel to each other, the outer and the inner membrane. So this is the outer one and this is the inner one. They will be arranged parallel to each other with a space in between them. Okay, a space will be there in between them and that space is called the perinuclear space. Okay, and it forms a barrier between the materials present inside the nucleus and that of the cytoplasm. So the nuclear membrane will act as a barrier between the materials which are present inside the nucleus and outside the nucleus, that is in the cytoplasm. Okay, so we can say the nucleus is surrounded by two membranes which form the nuclear envelope. It is surrounded by a nuclear envelope which is formed of two membranes. Okay. Now when we see the nuclear membrane, as I told, it is having an outer membrane as well as an inner membrane, isn't it? Both together forms the nuclear envelope. Okay. The outer membrane, it remains in continuation with the endoplasmic reticulum. Okay, the outer membrane will be in continuation with the endoplasmic reticulum. You can see in the picture itself, you can see that. Okay, so you can see here the outer membrane is in continuation with the, here this is the outer membrane is in continuation with the what? Endoplasmic reticulum. Okay, and they also bear ribosomes on it. What is present on the outer membrane? Ribosomes are also present on the outer membrane. Okay, and at many places in the nuclear envelope, it is interrupted by minute pores. Small pores are present in the nuclear envelope at many places, which are formed by the fusion of the two membranes. So wherever the outer and the inner membranes fuse with each other, there a pore will be formed, and that pore we say it as nuclear pore. So you can see many such pores are present on the surface of the membrane, or on the surface of the nuclear envelope. Okay, so the nuclear envelope is interrupted by minute pores, and actually these pores are formed by the fusion of the two membranes. Okay, then what is the use of this nuclear pores? The nuclear pores are the passage through which movement of RNA and protein molecules can take place in both directions between the nucleus and the cytoplasm. That is, the RNA will be synthesized inside the nucleus. So that RNA can move out of the nucleus into the cytoplasm through this nuclear pore, where it can help in protein synthesis, where it can be involved in protein synthesis. Okay, so the movement of RNA or any other protein molecules can take place in both directions. Both the directions means from the nucleus into the cytoplasm, or from the cytoplasm into the nucleus. Okay, that is the main function of nuclear pores. Okay, then normally there is only one nucleus per cell. In one cell, normally one nucleus will be there, but in certain cells we can see more than one nuclei also can be seen. Okay, for example, if you take the striated muscle, we can see more than one nuclei will be there. Okay, and in some of the cells, for example, the mature RBC, if you take, they do not have any nucleus. Okay, the mature RBCs, our own mature erythrocyte or the RBCs, they do not have any nucleus. In order to accommodate more amount of pigments inside it, that is hemoglobin inside it, this they will be lacking the nucleus. In the early stage, they will be having nucleus, but in the later stage, the erythrocytes or the RBCs do not have the nucleus. Okay, then the sieve tube cells, which are present in the vascular plants, they also will be lacking the nucleus. Okay, inside the nucleus, the matrix which is present is called as the nucleoplasm. Okay, nucleoplasm is the nuclear matrix, and this nucleoplasm contains nucleolus and chromatin. Okay, the nucleoli, it contains what? Nucleolus and the chromatin, and this nucleoli are spherical structures which are present in the nucleoplasm. Nucleoli is plural and nucleolus is singular. Okay, so this nucleoli are the spherical structures which are present in the nucleoplasm. The content of this nucleolus is in continuation with the nucleoplasm, because the nucleolus is not surrounded by any membrane. Okay, this nucleolus is not surrounded by any membrane. So the content, whatever is there in the nucleolus, will be in continuous with the nucleoplasm. Okay, and what is the function of this nucleolus? It is the site of active ribosomal RNA synthesis. So it helps in the synthesis of rRNA. What is rRNA? It is ribosomal RNA. Okay, so for the synthesis of rRNA or ribosomal RNA, this nucleolus is very much helpful. Okay, larger and more numerous nucleoli are present in cells which actively carry out protein synthesis. Okay, for protein synthesis, what is essential? Ribosomes are essential. For ribosomes to be formed, ribosomes, I told you, it is formed of rRNA and protein. Okay, so what is that rRNA? It is ribosomal RNA. So the ribosome is formed of ribosomal RNA. So wherever the cells are actively taking part in protein synthesis, they need more amount of nucleoli, more number of nucleoli, so that more amount of ribosomal RNA can be synthesized. Okay. We have seen that in the interphase nucleus, a loose and indistinct network of nucleoprotein fibers called chromatin is present, isn't it? Extended elaborate network of nucleoprotein fibers called chromatin is present. But when a cell is dividing, Okay, when a interphase is non-dividing phase, but when a cell begins to divide, the cells will be showing structures called chromosomes. In place of that nucleus, instead of chromatin, what will be seen? Chromosomes can be seen. Now, what is the difference between this chromatin and chromosome? Chromatin will be long, extended, elaborate network of nucleoprotein fiber. Then what is this chromosome? It is the condensed form of chromatin. Okay, what is it? It is the condensed form of chromatin. That means the chromosomes will be shortened. It will be coiled and it forms short and it becomes short, and now it is said to be as the chromosomes. Actually, both are having the same functionally, but only when we see the size of it, okay, it will be differing. Okay, the chromatin will be seen as long extended network of fibers, but chromosome will be the condensed form of chromatin. Okay, and chromatin will be seen in the non-dividing cell and chromosomes can be seen in the dividing cell. So this is the difference between chromatin and chromosome. Okay, the chromatin contains DNA and some basic proteins called histones. So which protein is the histones are there? Okay. In addition to this, some non-histone proteins and RNA are also there in chromatin. So what are the structures present or which forms the chromatin? So the chromatin contains DNA, some basic proteins called histone, then some non-histone proteins and also RNA. Okay, and if we see a single human cell, it is having an approximately 2-meter long DNA. So the DNA can be seen as a long thread of about 2 meter, which is seen in our human cell. Okay, and this DNA is distributed among the 46 chromosomes which is present in our cell. Each cell in our body contains 23 pairs of chromosomes or 46 chromosomes. Okay, so inside that chromosomes the DNA are present, and when we join one DNA with another, it will be extending about 2 meters in length. Okay, then when we see the chromosome, you are able to see the chromosome in the picture. Okay, the chromosome has a primary constriction or the centromere. So here you can see the primary constriction. Okay, so in this diagram you see it. So here you can see the diagram. Okay, so if you see here, you can see this is the primary constriction. Okay, constriction means that region is constricted, isn't it? Okay, so each chromosome is having a primary constriction which is said to be as centromere, primary constriction or the centromere. Okay, and on the sides of the centromere you can see the disc shaped structures which is called the kinetochore. You can see the kinetochore where it is labeled. Here you can see the disc shaped structure. Okay, I hope you are able to see that. Okay, so every chromosome is having a primary constriction or the centromere, and on the sides, on both the sides, what is present? Disc shaped structure called kinetochores are present. So what is the use of the centromere? The centromere will be holding the two chromatids of a chromosome. Okay, so this is the chromatid, this is the chromatid. So both these chromatids can be held by the centromere. So the centromere can hold two chromatids of chromosome. Here actually you can see how many chromatids are there. 4 chromatids are there. That is when you see when the cell divides at a later stage, you can see it will be dividing into what? Each chromatid will be dividing into 2. So 4 chromatids can be seen. Okay. So the function of centromere is to hold the chromatids of a chromosome. Okay. Based on the position of the centromere, the chromosomes can be classified into 4 types. What are they? metacentric, submetacentric, acrocentric and telocentric. What is metacentric chromosome? Here the centromere is present in the middle, so that it will be forming two equal arms of the chromosome. So in the picture also you can see here the centromere is present where? In the center, in the middle. Okay, so when it is present in the middle, it will be having two equal arms. On either side it will be having two equal arms. Okay, so this is said to be as metacentric. Meta here means middle. Okay, so this is the metacentric chromosome with the centromere in the center, so that the arms will be of equal length. Okay, now when we see the submetacentric chromosome, here the centromere will be slightly away from the middle of the chromosome, which results in one shorter arm and one longer arm. You can see here this is the longer arm and this is the shorter arm. So where is the centromere present? It is slightly away from the middle of the chromosome. Okay, so this type of chromosome is said to be as a submetacentric chromosome. Okay, now when we see about the acrocentric chromosome, the centromere is close to its end, forming one extremely short and one very long arm. Okay, so if you see here in this picture, okay, here you can see the acrocentric chromosome. So what is acrocentric chromosome? So here you can see the picture, acrocentric chromosome. What is it? It is having the centromere where towards close to its end, okay, towards one end, forming one very long arm and another a very short arm, very short, extremely short arm. Okay, and this is said to be as acrocentric chromosome. Can you follow? So what is acrocentric chromosome? The centromere is situated close to one of its end, forming one extremely short and another a very long arm. Okay, then what is telocentric chromosome? It is having a terminal centromere. That is, the centromere will be at one end. Okay, this is said to be as a telocentric. So this side there is no arm, isn't it? Only one side we can see the arm. Okay, see, if you see your 11th textbook, in that a short arm will be shown. Actually, it will not be having a short arm there. Okay, the centromere will be present at one end of the chromosome. Okay, so that is said to be as a telocentric. So based on the position of the centromere, the chromosomes can be four types, can be of four types. They are metacentric, submetacentric, acrocentric and telocentric. Sometimes a few chromosomes have non-staining secondary construction at a particular location. Okay, and this gives the appearance of a small fragment, and that small fragment is called a satellite. In the picture you can see that, isn't it? Yes, so what is that? In some of the chromosomes, what is present? A non-staining secondary construction is there, and this secondary construction is present at a constant location, and it gives the appearance of a small fragment in it, and this small fragment is called the satellite. Okay, so the chromosomes with the satellite, we can say it as satellite chromosomes. Okay, I hope you might have understood about this chromosomes. Next is the microbodies. So in both plant and animal cells, we can see many membrane-bound minute vesicles which are called as microbodies. Okay, what is present? Minute membrane-bound vesicles, sac-like structures are present in both the plant and animal cells, and these are named as microbodies. Okay, and normally they contain various enzymes in it. Okay, the microbodies contain various enzymes in it. Okay, so this is about the microbodies.